Good morning. I am A. Devasena. In this lecture, we are going to discuss about blood cell counter. This topic is present in the subject Medical Electronics. Blood cell counter. Blood is collection of specialized cells suspended in straw-colored liquid called plasma. Blood delivers oxygen and nutrition to body cells, collects waste, distributes hormones, spreads heat around the body to control temperature, and plays a part in fighting infection and healing injuries. Blood forms about 1/10 of the body weight of an adult, amounting to 5 liters in volume. Now we see about blood plasma. Roughly 50 to 50% of the blood plasma consists of the liquid only portion in which cellular components are distributed. Plasma is 90% water containing dissolved substances such as glucose, there is blood sugar, hormones, enzymes, and also waste products such as urea and lactic acid. Plasma also contains proteins such as albumins, fibrinogens, and globuloproteins or globulins. Alpha and beta globulins help to transport lipids, which are fatty substances such as cholesterol. Gamma globulins are mostly the disease-fighting substances known as antibodies. Let us now discuss about the specialized cells in blood. The remaining 45 to 50% of the blood is made up of three types of specialized cells. They are red blood cells or erythrocytes, which carry oxygen, various white blood cells known as leucocytes are part of the defense system, and the cellular fragments, which are also called as platelets or thrombocytes, which are involved in the process of blood clotting. Various specialized blood cell representation is shown below. In this diagram, the cross-sectional view of the blood cell is shown. In this blood vessel, we have seen the red blood cell, white blood cell, plasma, platelets. Now we see about red blood cells. Red blood cells are also known as erythrocytes. They are round discs with a diameter of 8 micrometer. Cell nucleus is absent in red blood cells. 1 cubic millimeter of blood contains about 4.5 to 5.5 million RBCs. The rate of RBC generation is 9,000 per day and its lifetime is 120 days. RBC is generated at the bone marrow. It has an iron-containing protein pigment called hemoglobin. It binds the oxygen molecule to the hemoglobin and transports oxygen through the blood. The oxygen-rich atrial blood is bright red in color and the oxygen-depleted venous blood is dark red in color. Now we are going to discuss about the role of hemoglobin, which is present in the red blood cell. Each red blood cell contains 300 millions of hemoglobin molecules. Hemoglobin is composed of heme, an iron-rich pigment, and globins, ribbon-like protein chains. Oxygen in the lungs latches onto heme to make oxyhemoglobin. In this conjoined form, oxygen travels through the bloodstream to all parts of the body. Red blood cell representation is shown below. Now we discuss about white blood cells or leukocytes. White blood cells are immune system cells that protect the body against diseases and foreign endeavors. While they vary in size, they are generally largest cell type found in the blood. It has an average diameter of about 10 micrometer. 1 cubic millimeter of blood contains 6,000 to 10,000 WBCs. The lifetime of WBC is 7 to 14 days. White blood cells reside throughout the body, including in the lymphatic system and in the blood. It helps to maintain the immune system of the body and fights against antibodies. White blood cell representation is shown below. Platelets are thrombocytes. The blood platelets is also called as thrombocytes. They are the mass of protoplasm. They have the diameter of about 2 to 4 micrometer. Platelets are tiny blood cells that help your body from clots to stop bleeding. If one of your blood vessels gets damaged, it sends out signals to the platelets. They form a plug, that is the clot, to fix the damage. Now we are going to discuss about thrombosis, that is blood clot. Thrombosis is the formation of blood clot known as thrombus within the blood vessel. It prevents blood from flowing normally through the circulatory system. When blood clot forms in the veins, it is known as venous thromboembolism. This can cause deep vein thrombosis and pulmonary embolisms. So far we have discussed about red blood cell, white blood cell and platelets. Now we are going to discuss about the function of blood cell counter. Changes in the normal functioning of an organism are often accompanied by changes in the blood cell count. The determination of the number and size of the blood cells per unit volume provides valuable information for accurate diagnosis. Blood consists of corpuscles suspended in a fluid called plasma in the proportion of 45 parts of corpuscles, that is cells of plasma. The percentage of cells in the blood is called the hematocritic value or packed cell volume (PCV). The majority of the corpuscles in blood are red blood cells, that is erythrocytes, other being white blood cells, that is leucocytes, and platelets, thrombocytes. By spinning blood in a centrifuge, the blood cells can be sedimented. The blood plasma with the blood cells removed is a slightly viscous yellowish liquid that contains large amounts of dissolved proteins. One of the proteins, fibrinogen, precipitates in the process of blood clotting and forms thin fibers called fibrin. The plasma from which the fibrinogen has been removed by precipitation is called blood serum. Blood clotting can be inhibited by the injection of heparin, a natural anticoagulant extracted from the liver and lungs of cattle. A count of the blood cells, an inspection of the size and shape, or a chemical analysis of the blood serum can therefore provide an important information for the diagnosis of diseases. Similarly, other body fluids, smears, and small samples of live tissue obtained by a biopsy are studied through the techniques of bacteriology, serology, and histology to obtain clues for diagnosis of diseases. So far we have discussed about the blood cell counter. Now we are going to discuss about the analysis done by the blood cell counter. First one, mean cell volume, that is MCV. It is calculated from the packed cell volume, that is PCV, and the number of red cells present per liter of blood. It is indicated in F/L, that is femoliters 1F/L is equal to 10 power minus 15. Then we are going to calculate mean cell hemoglobin, MCH. It is calculated from the HB and red cell count. It is expressed in picograms. Then the third one, mean cell hemoglobin concentration, that is MCHC. It can be calculated if PCV HB per DL are known. Now we are going to discuss mean platelet volume, MPV. It is the ratio of the integrated platelet volume to the platelet count and is expressed in femoliters. The next one is platelet crit. It is the percentage of the total specimen volume occupied by the platelets. Now we are going to discuss red cell distribution width, that is RDW. It is a numerical expression of the width of the size distribution of red cells. Now platelet distribution width, PDW. This index is related to the size range covered by those platelets lying between the 16th and 84th percentile.